Welcome back to another video of Pulsating Biology, and we were covering the chapter Nutrition in Plants. We've already covered few of the topics in the previous videos. Let's discover more about nutrition in plants. Since we have talked about how the food is prepared by the process of, or you can say the glucose is prepared in the plant by the process of photosynthesis, we are further going to learn that how the food that is prepared by the process of photosynthesis is basically digested and assimilated. So let's talk about how the digestion in plant and the assimilation in plant takes place. Now, digestion, by the term digestion, we mean that the macromolecules is broken down into micromolecules, and by the term assimilation, we mean that there is absorption of certain amount of substances. Let's talk about them in detail. So the number one pointer tells us that the starch that is stored is basically converted into maltose by an enzyme that is known as diastase. So the starch that is stored in the plant body is basically converted into maltose in the presence of an enzyme that speeds up a chemical reaction, and it is named as diastase. These pointers are important for your MCQs purposes. While the second pointer tells us that the maltose is converted into further glucose by an enzyme that is called as maltase. So the maltose, which is already converted from starch, it is converted into glucose by a presence of another enzyme that is named as maltase. These enzymes functions very appropriately in certain reactions. For the third pointer, we say that the glucose is either used in the respiration for energy or it is converted into sucrose. So when you talk about glucose that is formed after the conversion of maltose, it is used for the two purposes. Either it is used for the energy that is required for the process of respiration, or it is converted into further form that is known as the sucrose. When you talk about assimilation, the protein is basically converted into or digested into polypeptide and then to amino acid by enzyme that is known as the pepsin. So what happens is that the protein is converted into or digested into polypeptide, which is the form of a further protein that is further digested into amino acid with the presence of an enzyme that is known as the pepsin. Pepsin is an inactivated enzyme. It becomes active and converts the polypeptide into the amino acids. We have talked about the protein. Let's talk about the fat. So whenever we talk about fat, basically fats are broken down into two further processes. Either it is broken down into fatty acids or it is broken down into glycerols. So these are the pointers that you need to know when it comes to the digestion and assimilation of plants, that the starch is converted into maltose by presence of an enzyme that is a diastase. The maltose is converted into further glucose by presence of an enzyme that is known as maltase, and glucose is further used for the two purposes. Either it is used for the energy for the process of respiration, or it is used converted into the another form that is known as the sucrose. When you talk about protein that comes to the assimilation process, the protein is converted into polypeptide and then to amino acid by the presence of an enzyme that is called as the pepsin. And when you talk about the fats, so fats are converted into two further processes. Either they are converted into fatty acids or they are converted into glycerol. This is all that you need to learn about digestion and assimilation in the plants. Alright, let's talk about the main structure of a plant, that is the leaf. So when you basically observe a leaf, this is what a leaf looks like, and it consists of certain veins. This part of a leaf is known as the lamella. So what is the purpose of a leaf? So leaves are basically the food factory. This is where the photosynthesis process takes place by the absorption of sunlight. This is where the food factories are present and where basically the storage of food occurs. When you talk about the structure of a leaf, so the structure of a leaf consists of firstly lamella. Lamella is an outer surface or the leaf blade, you can say, that is present in almost all the leaves. That is you can call it the leaf blade. In the structure of a leaf, we have the petiole also. When you talk about petiole is the stalk of the leaf. When you observe the leaf, there's a little stalk that comes out of the leaf. That stalk is basically the petiole that is attached to stem, which continues in the lamella as a midrib. So this is the second structure. It actually basically what it does is that it holds the leaf out and erect to obtain more amount of light during the process of photosynthesis. If the petiole is absent, then the leaf have larger lamella. So for say if there is no presence of a petiole, so there would be a larger lamella and a shorter petiole. But if you say there's a longer petiole, then the leaves tend to be shorter. So the purpose of petiole is basically that it obtains more amount of light in order to make a leaf blade more prominent towards the sunlight. Okay, let's talk about the venation. When you talk about the venation, that means the presence of veins that are present in the leaf. Veins carries water. So what is the purpose of veins that are present in the leaves? They basically carries the water and minerals to the cells in the lamella. Inside the lamella, there are certain amount of cells that are present. So the water that is provided to those cells is through the lamella. So it's through the veins. So they carry water and the minerals to these cells in the lamella. The veins are connected to the midrib. These all the veins that are present in the leaf blade, they are connected to the midrib. This is the midrib. So all of the leaves are connected to the midrib. It forms a network of vessels around all the leaves. Each vessel is basically termed as the vein. All right, let's talk about the internal structure of leaf. When you talk about the internal structure of leaf, you need to remember certain structures. Let's say you need to remember that how the internal or the transverse section of leaves looks like. So it consists of two major processes, that is the epidermis. Epidermis consists of two parts, that is the upper part and the lower part. It has a waxy covering over it that is known as the cuticle. These are the two very important structures when you talk about the internal structure of leaves. So the internal structure of leaves consists of epidermis that contains the upper and the lower part, and it has a cuticle that has a waxy covering over it, which minimizes the loss of water. So its purpose is to minimize the loss of water. These are the two steps. Regardless of these two structures, we need to talk about the mesophyll cells. So when you talk about the mesophyll cells are basically present in the epidermis. These mesophylls is characterized into two compartments that you're going to learn in the next. So the third structure that you need to learn on the internal structure of a leaf is the mesophyll cell. So they are characterized into the two compartments. The mesophyll cell consists of two kinds of further mesophylls, that is either the spongy mesophyll or it consists of the pellucid mesophyll. These two functions differently. Spongy mesophyll works as they're very loosely arranged and which possesses air spaces between them. So this arrangement is very loose and it consists of air spaces between them. Regardless of this, when you talk about the pellucid mesophyll, it contains compactly arranged cells. So when you come to the arrangement of these cells, they are very compactly arranged and they are near to the upper epidermis. Both of these cells carry numerous number of chloroplasts. So these function very efficiently when it comes to the numerous number of chlorophyll. So both of them has very numerous number of chloroplasts, which carry basically the green pigment that helps to absorb the light and also gives a green color to the leaf. So this is the importance of mesophyll cell. There are two kinds of mesophyll cell: the spongy cell and the pellucid cell. At the lower part of the mesophyll cell, there is the presence of another kind of the cell, that is the stomatal cell. These stomatal cells plays a very important role when it comes to the plant. These are basically termed as the specialized kinds of the cell, or they can also be termed as the guard cells. These are the small pores that allows the entrance of water from outside environment towards the inside environment of the plant. So the stomatal are also termed as the guard cells because they have the pores. The presence of pores helps them to function efficiently. Let's talk about how the stomata work. We have already discussed how the stomata is very important for the plant, since they are the specialized cells that allows the water to enter inside the environment of a leaf. So these are the two guard cells that are present in a similar structure in the plant, and they are bean-shaped guard cells which surrounds the opening that is known as the stoma. So these are the two bean-shaped cells. The shape is termed as the bean-shaped cells which surrounds the opening that is called as the stroma. These cells are the only cells to manufacture sugar by photosynthesis in all the epidermis cells. Thus, they possess chloroplasts. So, another very important characteristic of these stomata are that they possess chloroplasts and they can manufacture their own sugar. These are the 3 pointers that you need to remember when it comes to how these stomata works. It's work by manufacturing their own sugar molecule, and by the presence of chloroplasts, they are able to help them to perform photosynthesis. When you talk about the two further functioning of these stomata, you'll get to know that they are either open or they are either closed. We are going to discuss how it works whenever they are open and how they works whenever they are closed. So whenever the stomata are open, basically they becomes very turgid. So let's discuss that when in the condition whenever they are open. So in the condition whenever the guard cells or the stomata are open, they consider or goes through a condition that is known as the turgidity, in which the cells become turgid and exerts pressure on the outer membrane of the leaf. So when the light increases, it increases the potassium ions inside the cell. So in the inner environment of the cell, the potassium ions increases. It lowers the water potential, and water starts moving out into the sac vacuole, which swells up. And due to the thick membrane in and the inner side of the guard cell, it basically curves. So what happens is that due to the turgidity, this plant cell or this tomato cell that is present inside the leaf basically swells up, and due to the thick membrane at the inner side of the guard cell, it curves. That's why we can also observe that in the diagram of a stomata, most of the stomatas are curved in such a manner. The central pores opens and allows water to move inside. So basically this part is the central pores that opens that allows the water to move inside. But when you talk about the condition when these stomata are closed, we've already talked about whenever it is open, but when you talk about the stomata condition when the stomata are closed, so it goes to a condition which is known as the opposite of turgidity, that is the flaccid, which is also known as the shrinkage. So whenever there is a shrinkage of cell, instead of becoming turgid, we say that the stomatas are closed. At night, usually this happens when all the sugar is used up. That means that the potassium ions that were inside the plant cell are used up at the night. Its water potential increases and it loses water. The cell shrinks and closes the pore. So whenever the potassium ion moves inside the cell, the sugar potential increases, and the water potential also increases. It loses the water eventually, and cell becomes shrink. This is how the closing and the opening of stomata basically works.